In the first part of how cells divide, we started talking about general process of cell division with prokaryotic cells, that being binary fission, and the purpose behind cell division. Why do cells divide? What is the ultimate end goal for that? Ideally, it's to produce an identical copy, but mistakes do occur, and we talked a little bit about the mistakes in the prokaryotic world, how that can lead to resistant strains of bacteria like MRSA, tuberculosis, etc. We'll also talk about what happens in eukaryotic cells if they fail to divide properly. But what we want to take a look at here is what's called the karyotype of a eukaryotic organism. And in this scenario, we're looking at an organism on the left-hand picture, an organism that has 23 pairs of chromosomes, that being human. So you get chromosome pair 1, 2, 3, all the way through 22, and then you have a chromosome pair called X and Y. Now this would be the karyotype of a human male, because it has an X and a Y chromosome at the 23rd pair, or what we call the sex chromosomes. If it was a female, it would be XX. When we look at some of these other examples, 39 pairs of chromosomes, or 24 pairs of chromosomes, or however many, that gives us an indication of the species that we're looking at. So chromosome numbers are a good indication of the species, fairly unique to the species. The information within the chromosomes is what gives us variation within the species. So all human males should have 22 pairs of chromosomes, and then the additional XY pair. So 23 total, but that's what makes you a human male. The difference between a male with brown hair versus blonde hair is the information found on specific chromosomes. So an organism, or at a point of an organism's life when it is haploid, we say it has one set of chromosomes. So haploid is represented by a lowercase n, and you have one set of chromosomes. When it is diploid, it's represented by 2n. This is when you have twice the number of chromosomes. So what we're looking at right here in these images, that is the diploid number of chromosomes. So 23 pairs represents the diploid number. If we ripped it in half to 23 individual chromosomes, then we would have the haploid number. So as we go through the cell cycle, the cell starts, in the case of humans, the cell starts with 23 pairs, but then it's got to get ripped in half to 23 individual chromosomes, 23 single chromosomes, and then the information gets doubled before the cell finishes and progresses to the cell cycle a second time. Right? Alright, so chromosome structure and some terminology is going to be important to be comfortable with here. So a chromosome is that little, what looks like a blue line with a green dot in the center, or the red line with a green dot in the center. You and me and everybody else on this planet inherited one chromosome from mom, one chromosome from dad. Mom's chromosomes came to us in the egg. Dad's chromosomes came to us in the form of the sperm. That combined created the zygote, which then went through cell division to make more and more and more copies to eventually produce the individual. Now, the little green spot on the chromosome is called a kinetochore. This is an anchor point for spindle fibers during cell division. So we'll get into that as we move through the process of cell division. So the chromosome on the left from mom should be identical to the chromosome on the right from dad, or should be fairly close, and we'll call them homologous chromosomes. Now, after they go through replication, then we have two blue lines, or what appear to be a blue X, and those are going to be called sister chromatids, because the blue X should replicate it, or the blue The blue line should replicate itself and then form the blue X. The red line should replicate itself and form the red X. The X-like structures are also referred to as homologous chromosomes, or you could refer to them as sister chromatids. Okay, so terminology gets a little goofy, but we want to try to make sure we can keep it straight, and as we go through the process of cell division, I'll try to re-emphasize the terminology and how it applies to each particular structure. So this is an overview of the eukaryotic cell cycle. Make sure you're comfortable with this. What are the order of phases? What's going on in each phase? What's the process, etc. That's why there's a red star there. Now, a cycle means it continuously circles and loops and just continuously goes around and around and around through that overall process. So you can start anywhere. Eventually you're going to wind up back at that same point. So for our discussion at this point, we'll briefly talk about G1, S, and G2, collectively known as interphase. Now, if this whole loop here is 24 hours, G1, S, and G2 collectively add up to probably 16 to 18 hours of the cell's life if it's a 24-hour lifespan or a 24-hour process. So things like your skin cells and your hair cells, it's roughly 24 hours for them to go through this. So if you're reading this lecture on a Wednesday evening at 7:30, by Thursday at 7:30, one skin cell has divided into two skin cells divided into 4, and so on, 24 hours. But the vast majority of that cell cycle is, again, 16 to 18 hours, is sitting in G1, S, and G2, or again what we call interphase. So during G1, the cell's going to grow, and it's going to make copies of its organelles, the mitochondria, the Golgi, the lysosomes, etc. When it goes into S phase, it's going to double its DNA. It's going to take the chromosome number and double it. When it goes into G2, it's going to synthesize specific proteins that are necessary for cell division. So critical things are happening, but looking at the cell, you wouldn't see much. It's not going to look like anything is going on at all. Now, once a cell enters prophase, this is when all the physical activity of the cell happens. This is when you can really see and notice changes. You'll watch a cell in prophase and be like, wow, I can really see things occurring. Prophase then moves to prometaphase. Prometaphase moves to metaphase. That goes to anaphase. That goes to telophase. And then the last portion is cytokinesis. Now, definitely something to point out. Cytokinesis does not begin after telophase ends. It's not like telophase stops and then you go to cytokinesis. Cytokinesis actually overlaps. It starts during the middle, roughly the middle of anaphase, and then overlaps through anaphase, telophase, and then kind of signals the end of the cell cycle when cytokinesis is complete. So from prophase all the way through cytokinesis is often referred to as the M phase of the cell cycle. As I mentioned earlier, the rough length of the cell cycle for skin and hair is 24 hours, but you get into cells like liver, different organs, different tissues, and it may take days or weeks or years in order for that cell to go through the cell cycle once. So if the cycle is meant for growth, healing the body, building new structures, repair, etc., if it takes you a year for one cell to turn into two into 4, it's going to take a very, very long time for that structure to heal if it's made up of cells that move very slowly through this process. Again, if you think about it, when you cut yourself, cut your hand, cut your leg, cut your face, wherever, it's not long before it's healed. Those cells are going through it very quickly. But if you break a bone, it takes a lot longer. Some cells, like nerve cells, don't ever really finish the cell cycle. They never really go through that cycle. And that's why when we destroy or damage nerve tissue, it's permanent. The body, that tissue typically does not heal. So consider spinal cord injury, depending on the severity of it and which nerves are damaged, you're not walking again. So things we'll talk about as we continue through the cell cycle. Okay, again, get familiar with this. Promise you'll see it again on a quiz or on an exam. So let's progress into the beginning of the cell cycle, or this entire process, and look at interphase. So key things during interphase. Cell grows, it replicates its DNA, and then it synthesizes or builds proteins and prepares for division. So it's kind of assembling everything and saying, hey, we got to get ready, we got to make sure we're getting everything put together, and that we're going to be ready for this cell division process that's going to start soon. But getting ready takes 16, it might be even up to 18 hours in order for it to get ready to actually start the division process. So mitosis is a window that is subdivided into smaller phases. And the first one that we want to look at here is called prophase. Pro means first. It is the first phase of mitosis. It is not completely separate from the cell cycle. It's just a phase of the cycle. So key things I want you to know in the cell cycle during prophase. The nuclear envelope start to break down. These little structures called centrioles, those little green star-like things there, they're also called asters. Those get assembled. Those are protein-based structures. They will get assembled and built during prophase as the nuclear envelope's disintegrating and breaking down. At the same time, the chromatin, which is the genetic material, the DNA, is going to coil and kind of spin and wrap itself around those proteins, creating the structure we call the chromosomes. So chromosomes actually become visible during prophase. They look like these little X's. So you'll see the slide we had before back in interphase, the nucleus contained what looked like just a bunch of string and like a big pile of spaghetti noodles. Once the cell's into prophase, it looks more like little X's or little individual threads. So also keep in mind these phases are continuous. It's not as if prophase starts immediately at a certain time, it finishes immediately. They just kind of blend and transition into the next phase of the cell cycle. So we've got interphase, big things happening there in regards to copying your DNA, the cell growing, making proteins, and then now we march our way, or kind of transition our way into prophase. Nuclear envelopes breaking down, chromatins condensing into chromosomes, spindles are forming, or we call them centrioles and asters. Same structure. Those are all forming, and everything is getting ready to then move into the next phase.